Meiosis, the cell's wildest party. What even is meiosis? All right, let's start simple. Meiosis is what happens when a cell wants to make gametes, sperm or egg cells. Think of it as DNA's version of less is more. The goal? Cut the chromosome number in half so when sperm and egg unite, things don't get overcrowded. Without meiosis, every generation would double their chromosomes and we'd all look like walking science errors. Meiosis equals order in the genetic chaos. Boom. The first meiotic division, the crossover episode. So here's where the action begins. The first meiotic division is all about reducing the chromosome number by half. It's like downsizing your stuff before moving into a smaller apartment. Painful but necessary. The cell starts with a full set of chromosomes and ends up with 2 half-loaded cells, each still carrying unique DNA. Let's break it down stage by stage. Process 1. DNA Tinder edition. Chromosomes pair up with their lookalikes, homologous chromosomes. They cozy up so close that bits of DNA literally swap places, crossing over. It's like trading sneakers with your best friend. Same style, new flavor. The spindle forms, the nuclear wall disappears, and suddenly it's chaos, but productive chaos. Diversity is born right here. Metaphase 1. Lineup time. All the chromosome pairs line up neatly across the middle of the cell, holding hands like contestants in a game show. The spindle fibers attach, ready to pull them apart. The random lineup means no 2 cells will ever be identical. That's why siblings can share parents, but look like they came from different franchises. Anaphase 1. The great pull apart. Spindle fibers yank each homologous chromosome to opposite poles. It's like a dramatic movie breakup. You go north, I'll go south. Sister chromatids stay together for now, just vibing. The tension's real. Telophase 1. Split decision. Chromosomes reach the poles, the cell pinches in the middle like it's splitting the bill, and bam, 2 new cells appear. Each one has half the original chromosome set. Congrats. You just went from diploid to haploid. It's official. You're halfway to making gametes. The second meiotic division, the sequel nobody asked for. Think of this as the cleanup round. The first division cut the chromosome number in half, and now the second one separates the sister chromatids. Basically, the cell making sure each new cell gets its own copy. It's like when a band breaks up and every member goes solo. Let's walk through the encore. Prophase 2. Round 2, no repeats. No rest for the weary. Those 2 cells from before dive straight into another division. The spindle forms again, nuclear membranes dissolve, and chromosomes gear up to move. Déjà vu, but faster. It's like the sequel that didn't need to happen, but somehow makes the story complete. Metaphase 2. Line up again. This time, single chromosomes, not pairs, line up at the equator. They're like solo artists now, ready to go their separate ways. The spindle fibers attach to each chromatid centromere like paparazzi snapping shots. Anaphase 2. The final split. Boom! The spindle fibers shorten and drag sister chromatids apart to opposite sides. Each half now has 1 copy of every gene combo made earlier. It's organized chaos, biology's version of spring cleaning. Telophase 2, the grand finale. The chromosomes finally chill at opposite poles, nuclear envelopes reform, and the cell divides again. The result? 4 brand new cells, each genetically unique, each with half the DNA. That's 4 little potential sperm or egg cells, nature's personal lottery tickets. Why it matters and why it's awesome. Without meiosis, we'd be carbon copies of our parents, boring. Thanks to crossing over and random assortment, every human is a one-of-a-kind remix. Your freckles, your dimples, your oddly specific love for pineapple pizza. That's meiosis doing its magic. It's messy, dramatic, and absolutely brilliant. So yeah, meiosis, the cell's wild party where everyone leaves different but better for it.